My name's Christine.
My name's Arfa.
And my name's Amanda.
Our project is about a study done at the University of Colorado Boulder about the structure of histone-based chromatin in archaea. What are histones? What is chromatin? What is archaea? If you answered I don't know to any of the above questions, you're in luck. Let's break their study down. Archaea, bacteria, and eukarya are the three domains of life. Archaea is said to be the closest living relative to an ancient ancestor that underwent DNA folding. Both domains use histones with their DNA folding. The study showed that archaeal DNA folding suggested a divergence between the domains.
Histones are proteins that fold around DNA. Chromatin is a double-stranded helical structure of DNA. In eukaryotic DNA folding, chromatin first combines with histones to form nucleosomes. Each nucleosome consists of proteins around which DNA wraps. In order to form the structure of the nucleosome, eukaryotic DNA wrap around archaeal histones. This allows nucleosomes to pile up on top of one another to allow DNA to be tightly packed into small spaces and to regulate gene expression by allowing various regions of chromatin to interact with each other. Multiple nucleosomes eventually fold and compress to form a chromatid of a chromosome. This DNA folding method varies between archaea and eukarya. Eukarya use 4 different types of histones. They use 2 of each type of histone on each strand of DNA for a total of 8 histone molecules on each nucleosome. DNA strands are wound around these 8 histone proteins forming the nucleosome. Nucleosomes are strung together on a strand of DNA and eventually become chromosomes. In archaea, DNA is wrapped around one type of histone protein to make a superhelix.
Now that we know the general process of DNA folding in eukarya and archaea, let's discuss the experiment. The study was a series of tests to see how these histone proteins were behaving in DNA folding of archaea and eukarya. The scientists use X-ray crystallography, which bombards crystals with X-rays, to yield information regarding the position of all amino acids and nucleotides in the DNA complex. The researchers synthesized crystals of the histone DNA complex in the archaeal species Methanothermus fervitus. After inspection, the researchers were able to determine that DNA folding in archaea creates a similar structure to those found in eukaryotic nucleosomes. The results concluded that most features of eukaryotic compaction of DNA into nucleosomes is maintained in archaeal histone-based chromatin, suggesting a common ancestor between the two domains.
Each histone fold dimer in archaea interacts with the DNA in a similar fashion to eukaryotic histone dimers. All histones have a central histone fold dimerization. Histone fold dimers are fully conserved amino acid side chain interactions that multiple studies have shown are essential for DNA binding. Scientists also showed that the polymerization of eukaryotic histone dimers was limited by their asymmetry to an octamer, 8 different histones. The histones could only be in one specific position. In contrast, archaeal DNA forms an uninterrupted long superhelix with only one type of histone, almost like a continuous nucleosome. The histone involvement in the DNA of both archaea and eukarya is exactly the same. However, eukaryotic histone DNA complexes are limited to one compact structure of nucleosomes stacked on top of one another to form the chromosome. Many of the archaeal histone proteins recently discovered lack basic histone tails, which are specific to each eukaryotic histone and contribute to gene expression regulation and overall stability in the nucleosome.
To see if the extended polymerization in archaea had an importance, the researchers introduced mutations to destabilize the superhelix without affecting the DNA binding ability. The results showed that the superhelix had trouble growing under stressful conditions, wasn't using its genes properly, and had a delay in response to nutrient change. The purpose of this was to show that the archaea DNA folding is an early prototype of the eukaryotic nucleosome. A variable length chromatin structure was replaced by a defined nucleosome consisting of 4 distinct histones very early in eukaryotic evolution. This study relates to evolution and natural selection. With increasing genome size, it was necessary to limit histone assembly to defined nucleosomes to allow further compaction into organized but still readily accessible higher-order chromatin. Both eukarya and archaea have a common ancestor, but differences in DNA folding was the defining characteristic that led to 4 distinct organized histones being selected for. Archaeal histone DNA complexes aren't limited to a discrete structure, so their gene regulation differed from eukaryotes. Eukaryotes, over time, gained the ability to selectively position nucleosomes. This allowed them to evolve a more complex and organized way to fold their DNA. And that's about it. Thanks for watching.